In this video, let's talk a little bit about how the liver can act as a metabolic hub. Now, the liver is a very complex organ that has a whole variety of functions, but we're going to focus on its metabolic functions here. Now, the actions of the liver metabolically will fall into two main categories: storing nutrients during times of plenty in what we refer to as the postprandial phase after a meal, or releasing nutrients in between meals when those nutrient levels are falling due to use by our peripheral tissues. And so immediately following a meal, we would have nutrient uptake from the GI tract, nutrients such as amino acids and sugars and fats that would be leaving in the bloodstream that's draining out of that GI tract, but that blood is delivered to the liver first through a specialized circulatory pattern known as the hepatic portal system. And so these nutrients come in high quantities toward the liver in that postprandial phase. There they can be stored anabolically as glycogen in the case of carbohydrates, as triglycerides in the case of fats, or as protein in the case of amino acids. Now that's largely stimulated by, and it's more complex than this, but largely stimulated by both the nutrient levels and by the hormone insulin that's very high immediately after a meal. Now, between meals, the liver tends to act catabolically, breaking down these stored nutrients and delivering them for peripheral tissues to utilize. Tissues like the brain over here in the upper right, that's going to utilize glucose that's being released by the liver, can also use ketone bodies produced from triglycerides by fat metabolism. It tends to use more ketone bodies during periods of more prolonged fasting when fat metabolism rises. If we look in the upper left over here, we have our red blood cells. Our red blood cells are kind of unique in that they lack mitochondria as adult cells, and therefore cannot run the Krebs cycle or the electron transport chain. They simply aren't there. And so these cells rely entirely on glycolysis, converting glucose into lactate anaerobically, and to make ATP, but in the process they make this product lactate that has to be converted back into glucose. And that actually takes place at the liver as well, showing our lactate being converted back to glucose here. If we look at fat tissue down here in the lower left, fat tissue is also a storage site as well. Glucose uptake there can be converted to triglycerides into fat tissue. But during periods of fasting or in between meals, our triglycerides are broken down and we release those stored nutrients. And now we break down our triglycerides into their component parts, glycerol and the fatty acids. And then, of course, those can be utilized by muscle tissue and other tissues. The glycerol is interesting because in more prolonged fasting, that glycerol can act as a substrate for the making of new glucose. It's called gluconeogenesis. Once again, that is accomplished by the liver, where we're going to take these substrates from the adipose tissue, and as we'll see in a moment, from muscle tissue, to make glucose to supply for other tissues when we haven't had nutrient intake for a more prolonged period. In shorter periods, we have enough glycogen stored at the liver to maintain those blood nutrient levels, the blood glucose levels specifically. But in more prolonged fasting, we're going to use metabolites from adipose tissue and metabolites from muscle tissue to maintain that blood nutrient level. And so here we can see, here's our regular muscle metabolism where we're using glucose to make ATP, we're using fatty acids to make ATP to fuel those muscle contractions, but we also make lactate just as the red blood cells do. And again, that can get converted back to glucose by the liver. But in more prolonged fasting, when the hormones glucagon and then cortisol rise, the proteins in skeletal muscle will be broken down to release amino acids to supply to the liver to run that gluconeogenesis, maintaining our blood sugar levels, our blood glucose levels during prolonged fasting.